So in this module, I'll be teaching you guys how to analyze your data downstream in R. So the 16S data, the ASV table, and the taxonomy that you generated in the previous module, we're going to be importing into R and then doing some downstream analysis to interpret the data. So first off, let's get RStudio running from the Elifu Slurm cluster. Now if you go to Vula, there's some detailed instructions there, but I'll also run it through you. So firstly, we're going to log in to the cluster with SSH. So again, Gerrit, you'll just replace with your own username. So for myself, I'm just going to SSH onto that Elifu cluster. You'll see it says Slurm login now. And then what I'm going to do next is I'm going to start an interactive session. So I'm just drawing up the srun command that I used previously, and I'm going to copy and paste that so I can get onto a worker node. So now you see I'm on worker node 001. And now next thing you'll have to do is on your local machine, go to your .ssh/config file. If you don't have one, create one, and then you want to copy and paste this content into that file, and you'll be able to copy and paste that from the instructions on Vula. If I go to my own SSH file just to show you guys, so I'm going to open a new window. Now you can see, so in my case I've got my username and I've got all that information for this specific host. So if you'll just do that, and once you have done that, we can start an interactive job, which we've already done in this case. And after that, we're going to launch RStudio with this command over here, substituting your own username here, and then you're going to make yourself your own secure RStudio password. And what we're specifying here is the singularity container that contains everything that you need for this module and for running R downstream. So you already have the R packages installed that we're going to be using. So we're going to enter the RStudio password. You can make up anything, maybe in my case my underscore password, and then you're going to specify this RStudio container, and then it's going to give you a specific port number that you're running on. So you're going to need that port number. So the next thing we're going to do is from your local machine, then you're going to SSH onto that worker node. It might be 0001, it might be 0002, and then you're going to specify that number that it said it's running on that port at the moment. So if I start a new window from my local machine, so I was on 0001 and port 34025, 025, and now you can see it puts me on a worker node. So the next thing you want to do is from your browser, you're going to open this localhost here. And then it should bring up the RStudio login for us, and that'll allow us to log into RStudio. So for me, it's automatically logged in now. You will get a blank page here. I've already got the script loaded, but as long as you can open this RStudio on your local host, you're good to go. So the RStudio login box will look like this. So you'll enter your username and the password that you created, and then the blank RStudio screen will look something like that. Okay, so now we can move on to discuss the packages we're going to use and why we need them. So 16S microbiome data have specific characteristics. So it's count data, and that data has a skewed distribution often, and it's zero inflated, so that means there are a lot of zero counts in our data frame for samples where that specific OTU may not be present. And there are also differences in absolute recounts between samples, and therefore we're going to have to normalize all samples so we can compare between samples. And you may also have redundant taxonomic information. So you can have, maybe ASVs that are very similar. They might differ in a few base pairs, but they have the same species level annotation. So maybe they're slightly different strain variations, but from a point of view of biological interpretation, it's often useful to merge them down to a lower taxonomic level. Now, fortunately, there's been a lot of work done in R for 16S type data, and so there are specialized packages for analyzing our downstream data. So some of the ones that we're going to use are Vegan and phyloseq. They're very commonly used, and they're great for exploratory data analyses, especially plotting, and we're going to be doing that in the next session. And then also for differential abundance testing, there's a package called MetaGenomeSeq that we're going to be using that was specifically designed for analysis of sparse high-throughput sequencing data. So just before we get started hands-on, I'd just like to discuss the R package phyloseq, and specifically something called a phyloseq object. This is what we are going to use to store our data. So phyloseq uses a specialized system called the S4 class, and you store all related phylogenetic sequencing data together with your metadata as a single experiment level object. This is really useful for your downstream analyses because you can access different slots of this phyloseq object, and it might be a little bit easier to understand if we look at this graphic. So we'll have our OTU table, and we're going to import our sample data, so that's your maybe clinical data of interest, our taxonomy table, and then you can optionally also have a phylogenetic tree. And all of these objects form part of this experiment data phyloseq object. And that is really useful because it really prevents you from making errors maybe in sample names between your OTU table, your taxonomy table, and your sample data, so that you're sure you're not mixing up samples between those separate data matrices. So you can see there actually that your taxonomy table is a matrix, and then your sample variables are a data frame, and each one of these objects form part of this phyloseq object. So this will become a little bit clearer once we actually work with this phyloseq object. So if we try and import our data into R, what we're going to have to do is take the data2 output. So you guys should have saved it as .rds objects, and you should have an ASV table and your taxonomic annotation. And then we'll also have some metadata that we'll supply, and often these are in CSV or text format. So just before we get started in RStudio, you're going to need a number of files to do this analysis. So when you're on your home directory on the worker node, on the Slurm cluster, you can create a directory called r_downstream for yourself with the make dir command. And then in this directory, you should have the dog microbiome tutorial data2.r. This you can find on the Vula website. You should have the microbiome _customs functions _tutorial.r, and also the practice dataset metadata. So that's our sample data that we're going to import. And then you'll also have from the previous module, and we'll also upload them again, the .rds objects. That will be your ASV tables. In this case they're called ASVs underscore counts and underscore taxonomy for the ASV and the taxonomy tables respectively. So in a previous session we created a R project which made it easier for us to reference files such as this one without having to specify the full user path. So you can go and do the same now. So when you go to File, New Project, you can create that project and specify that R underscore downstream folder that we created to have your data in there. Okay, so once you've created your R project, you can load this dog underscore microbiome tutorial data2.r script file that we're going to be working from, and you can find that file, open file, and that should bring up your R underscore downstream folder that you created your project in, and you should find the script file that you need there. So we open that, and the next thing we're going to do is use the source command to import a collection of custom functions that we're going to be using in the next session. So we can look at that file. So again, we're going to say file, open file, and it's just another R script file, but in this case it's composed of customized functions that I've built up that are very useful for us downstream, and you'll see this is where we load all our R packages, and that's why for this tutorial script you don't see the library commands that you normally would in your script. So we'll look at this in more detail in the next session, and you're also welcome to go through it, but you don't have to know any of this in detail. But all it is a collection of functions. For instance, here we have the make metagenome-seq function that'll convert a phyloseq object to a metagenome-seq object so that the two packages can talk to each other. And what the source command does is it loads those functions into R's memory so that we can just call those functions by name and use them. Okay, so let's get started and import our data. So we're going to use the read RDS function to import that ASV counts file. And if we look at that, you'll see it's a matrix of over 4,000 ASVs for 15 samples. And what we're going to want to do, as you can see here, there aren't actually any ASV IDs like ASV 1, 2, 3 for our ASVs. They actually give you the whole sequence, which is good, but it's also not ideal for plotting because it's quite cumbersome. So we're going to replace those sequences, save them in memory, and give them user-friendly ASV IDs. So then if we look at the dimensions of this table, you'll see 4,415 rows. And then if we similarly import our taxonomy table and look at the dimensions of that matrix, you'll see also 4,415 rows. So it's good practice when you import this just to do a check to see that these rows are identical. And for that we use the function identical, and we just check the row names of our ASVs against the row names of our taxa. And in this case they are identical, so we get true, otherwise we would have gotten false. And in the next video session we'll continue from where we left here to create those ASV IDs.